everyone, this is the Theoretical Doctor and welcome back to my channel. If you want to access flashcards on this topic and other videos, do click the link below. I post weekly videos every Tuesdays and Fridays, so do subscribe for more content. Today we are starting a new subtopic on proteins and we will dive into amino acids and peptide bonds. Amino acids are the basic unit of a protein. And proteins are polypeptides formed from the condensation of amino acids joined by peptide linkages. So before they become proteins, they are, in tiny subunits. They're called the amino acids, and when they combine together through the process of condensation, they form polypeptides. Well, first dipeptides, then polypeptides, and the chain that connects them, it's the peptide linkages. And there are four classes of amino acids. You have the non-polar, polar, acidic, and basic. And the acidic and basic group falls under electrically charged amino acids, with the acidic being negatively charged and the basic group being positively charged. So this is the basic structure of an amino acid. So just practice drawing this. So first you have the carbon over here. This part is always hydrogen, and then on the side you have the amine group, and over here you have the carboxylic acid group, and the bottom part is a residual group, side chain. So that part will always change. And I mentioned earlier that there are four types of amino groups, four main classes, so you have the polar, non-polar, acidic, and basic, and it can be either of this. So just something to help you remember: polar ends with hydroxyl group, and non-polar is hydrophobic, so it ends with hydrogen, and acidic is carboxylic, and basic is basic amine. There is, variation in the side chains, and it's important for the intramolecular and intermolecular interactions, because that determines the structure and the functions of protein. I'll come back to this again at the end, but first let's take a look at the background of different groups of amino acids. So the first part is the non-polar group. It contains alkyl or aromatic side chain as its R groups, that is not electrically charged, so it's hydrophobic. They do not form hydrogen bonds with water. So you see here, it either ends with H or hydrogen, or it ends with CH3. As you can see here, glycine is the only one that ends with a hydrogen, whereas the rest all ends with CH3, so that falls under non-polar group. Now for the polar group, they have functional R groups and they're hydrophilic, so that means that they're able to form hydrogen bonds with water and take part in chemical reactions. And next you have the acidic and basic group. So these are electrically charged amino acids, and when they're electrically charged, they can either be acidic or basic. So acidic amino acids have side chains which contain negatively charged carboxyl group. So remember, acidic, carboxyl, negatively charged, because you can see here they're negatively charged at physiological pH. Now, basic amino acids have side chains which contains positively charged amino group. You can see here it's positively charged. So remember, basic amino base. I don't know how to actually make this easier, but I remembered while I was studying that was how I used to remember this. So I'll come back here. Just refer back to this table. So polar hydroxyl ends with OH, non-polar hydrophobic ends with H or CH3, acidic carboxylic ends with this, and also basic amine. Alright, so let's talk about the formation of peptide bonds. A dipeptide, di meaning two, is formed through the condensation reaction of the amino group of one amino acid and the carboxyl group of another amino acid linked by a peptide bond. So, okay, this one. Okay, so you have the carboxylic group over here and the amine group over here. So it is formed through the condensation reaction of the amino group of one amino acid and the carboxyl group of another amino acid. So when they come together through condensation reaction, condensation means removal of water, and they will form. So this part over here is the peptide bond, and because it's between two amino acid, you get dipeptide, and you'll form this. But as you keep adding more and more and more, it becomes a polypeptide, and poly meaning more. So a polypeptide is formed when more than two amino acids are linked together, and the process is called polymerization. Alright, so here you notice that as you keep adding from dipeptide, they become more and more and more. There is a repetition in pattern. So the polypeptide chain has a backbone of atoms in regular pattern, and it becomes like this, and CC and CC. You can try and practice and draw, and it will actually become like this. So after that, obviously, when you add it like this will become the side, or maybe this will be the other end. You'll notice that there is a free amino group at one end, and there is also a free carboxyl group at the opposite end. If you've reached this point, thank you very much. Do subscribe and share this video to those who might find this helpful, and I'll see you in the next one.